Hi everyone, my name is Giselle Hernandez. For this journal club, I chose to discuss an article about how the COVID-19 pandemic has influenced early life gut microbiome development. As we know, the pandemic changed many aspects of our lives, from the way that we interact socially to the way that we maintain hygiene. But what about its impact on newborns? So, to start off, for the bottom line up front and why we should care, I'd like to start by saying that the gut microbiome plays a crucial role in immune system development, and early life microbial exposure is key to shaping long term health. And the question that this study aimed to answer was, how did pandemic related hygiene restrictions affect the development of the infant gut microbiome? To investigate this, the researchers decided to study two groups of infants, one before the pandemic and one born during the pandemic. And then they analyzed stool samples using a technique called shotgun metagenomics that allows for a detailed examination of microbial composition and diversity. Before this study, we already knew that environmental exposure significantly influences gut microbiome development, and research had shown that children in urban settings where hygiene standards are higher tend to have a less diverse microbiome and thus are more prone to autoimmune conditions like allergies and asthma. Um, the COVID-19 pandemic introduced extreme hygiene measures because of the lockdowns, the social distancing, more sterilization, and this may have further limited microbial exposure. For instance, WASH was implemented, which stands for Water, Sanitation, and Hygiene, and it was this public health framework that focuses on providing access to clean water, proper sanitation, and hygiene practices to prevent disease spread, and it's crucial for preventing infections and maintaining public health, especially during pandemics. So the World Health Organization, they emphasized hand washing as a key strategy to prevent COVID-19 transmission, but access to wash services was limited in many low-income countries because vulnerable populations, they lacked clean water, soap, sanitation, and this made it difficult for hygiene recommendations to be followed. And then another interesting fact that I found from another outside source is that the overuse of hand sanitizer can negatively impact the gut microbiome by first disrupting the skin microbiome, which has a close connection to gut health. So, absorption through the skin can alter microbial diversity and could potentially lead to something called gut dysbiosis, which is an imbalance in gut bacteria that is linked to health issues. And so this study investigates whether these pandemic related behaviors had unintended consequences on infant gut health. Understanding these effects is crucial because early microbiome disruptions can lead to long term health issues. The gut microbiome helps train the immune system, and then alterations in its development have been linked to things like I mentioned, like allergies, or inflammatory bowel disease and metabolic disorders. So, if the pandemic hygiene measures negatively impacted microbiome development, this could have significant implications for public health policies and infant care recommendations moving forward. And this is something very important to consider because this is our next generation's health development. Now, looking at the article, the key research question they aimed to answer was, did infants born during the COVID-19 pandemic develop a different gut microbiome compared to those born before the pandemic? Um, and the researchers aim to determine whether these changes might have lasting effects on the immune system function and overall health. Now, looking at the methodology and the study design, to answer this question, like I mentioned, the researchers studied two cohorts of infants in Hong Kong. One was a pre COVID group, which was born between 2017 and early 2020, and the other was a during COVID group that was born between April of 2020 and January of 2022. So they collected stool samples at multiple time points, ranging from birth to 12 months, and then they used shotgun metagenomic sequencing to analyze microbial diversity, antimicrobial resistance genes, and functional changes in the microbiome. And this technique provided high resolution data that allowed researchers to compare the microbiomes in greater detail. However, usually with shotgun metagenomic sequencing, they use Illumina sequencing technology that we discussed in Module 1. And this method of sequencing allows researchers to sample genes in organisms in a complex sample to examine bacterial diversity and the abundance of microbes in chosen environments. Illumina sequencing provides high quality bases at a low price per base sequenced, and it has the highest throughput out of any platform or technology out there. However, this technology has the disadvantage that it has the shortest read length of any platform. And so assembling a genome from these short sequences is more difficult. So that increases the rate of error. And I found from an outside source that shotgun sequencing traditionally requires millions to tens of millions of high quality reads per sample, which is a lot compared to other technological ways of doing this. And this limitation can hinder the accurate identification of certain microbial species and detailed functional analyses. And it can lead to things like overrepresentation of abundant microbes. Now, getting into the results, let's look at what they found. The first major result that they found was that infants born during the COVID-19 pandemic had significantly lower microbial diversity, and we can see this in lower KO1 and Shannon Diversity Index scores, which are both measures of how many different types of bacteria are present in the gut. And the reduction in diversity was especially noticeable at 2 to 3 months, 6 months, and 12 months. Um, so, since microbial diversity is key to a resilient immune system, this suggests that pandemic related hygiene measures limited the natural microbial exposure that infants should normally be receiving. And I chose to analyze figure 2 from the article because it presents core evidence that microbial diversity was significantly reduced in the during COVID cohort. And so, to understand the study's findings, let's first go over the KO1 index. It's a metric that's used to measure microbial richness or the number of different species in a sample. And it's particularly useful because it estimates not just the species that were detected, but also the undiscovered or rare species that might be presented but missed because of limited sampling. And a higher index means more microbial species are present, which indicates a greater diversity in the gut of the bacteria. And a lower index suggests reduced microbial richness, which can be a sign of limited microbial exposure. The KO1 index results showed that microbial richness was significantly lower in the during COVID group at multiple time points. Like, for instance, from 2 to 3 months, the index was significantly lower with a p value of 0.027. Whereas if we look at 12 months, the trend continued with a p-value of 0.0028. And this tells us that infants born during the pandemic had fewer total microbial species in their gut compared to those born before the pandemic. While the KO1 index measures species richness, now looking at the Shannon index, we're looking at microbial diversity, meaning it considers both the number of species present and how evenly distributed they are. So the study found that Shannon diversity was significantly lower in the during COVID group. For instance, they saw that at 6 months, the p value was 0.004, whereas at 12 months, the p value was 0.007. And this means that not only were there fewer species overall, but the microbial communities were also less balanced, with some bacteria dominating while others nearly disappeared. Now, looking at figure 2C, we're looking at beta diversity, which, beyond just species richness and diversity, they wanted to examine the beta diversity or how the entire microbial community structure differed between the two groups. So, to do this, they used Bray Curtis similarity, which was used to quantify the difference between two samples based on their species composition. and principal component analysis, or PCA, which helps with the visualization and interpretation of the data. And what they found were significant differences between the pre COVID and during COVID groups again. At birth, there was a p value of 0.017, whereas at 6 months, there's a p value of 0.041. And these values mean that the overall composition of gut bacteria in infants was drastically different depending on whether they were born before or during the pandemic. So, what factors were responsible for these microbiome changes? The study identified the three biggest influences on infant gut microbiome composition, which were delivery mode, so whether the baby was born vaginally or via C section, which affects initial microbial exposure, the COVID-19 pandemic itself, which, as we discussed, increased hygiene standards, and intrapartum antibiotic use, or antibiotics that are given to mothers during delivery, which can alter bacterial colonization in newborns. And these factors combined to significantly reduce microbial richness and diversity in infants born during the pandemic. Beyond overall microbial diversity, the study found that specific bacterial species were impacted. Beneficial bacteria were reduced in the during COVID group, especially. Um, for instance, and pardon me if I mispronounce these, Bifidobacterium breve and Bifidobacterium dentium, important for digestion and immune function, were lower at 2 to 3 months, and Klebsiella pneumoniae and Klebsiella varicola, which are bacteria that help regulate gut metabolism, they were lower at 6 and 12 months. And the reduction of these beneficial bacteria could, in turn, weaken gut function and immune development in infants. Now let's look at the maternal microbiome. I chose figure 3, which analyzes four key aspects of the maternal microbiome. It analyzes in figure 3A microbial richness and diversity, again using the KO1 index, the gut microbiota composition in figure 3B. 3C looks at key host factors influencing microbiota composition, and 3D looks at differential bacterial species. So let's break down these findings one by one. For figure 3A, we're looking at maternal microbial richness and diversity. So the KO1 index and Shannon index were once again used to measure these factors. And the key findings were that there were no significant differences in maternal microbial richness and diversity between the pre and during COVID cohorts, because we can see a p value of 0.43 for KO1 and p equals 0.099 for the Shannon index. And this matters because, unlike infants who showed a clear reduction in microbial diversity, the mother's gut microbiota remained relatively stable, which suggests that the pandemic's hygiene measures did not drastically impact maternal microbiome richness. Um, then we're looking at figure 3B. Even though the overall microbial richness didn't change, the figure 3B shows that the composition of the maternal microbiome did shift significantly during the pandemic. Again, we're looking at principal component analysis based on the Bray-Curtis dissimilarity. And this revealed that the gut microbial community structures were significantly different between pre COVID and during COVID mothers. And this suggests that specific bacterial species increased or decreased in response to pandemic related lifestyle changes. Looking at figure 3C, we're looking at what could have possibly caused these changes. And here they identify the biggest factors influencing maternal microbiome composition. The first one being the COVID-19 pandemic itself. It was a major determinant which altered microbial composition significantly. Another major factor they found was smoking status, which had a strong impact on microbiota variation. And then other factors like age, gestational diabetes, and gestational hypertension had less significant effects. The key takeaway from figure 3C is that COVID related lifestyle changes like social isolation, reduced travel, and increased sanitation played a significant role in altering maternal gut microbiota composition. And lastly, for this figure, figure 3D, it identifies specific bacterial species that were significantly different between pre and during COVID mothers. Some notable changes were that several bacterial species were differentially abundant, including Faecalibacterium prausnitzii, Blautia wexlere, and Collinsella aerofaciens. However, these maternal microbial changes did not directly overlap with the species that changed in infants. This suggests that while maternal microbiomes were altered, these changes seen in infants were likely caused by reduced environmental microbial exposure rather than the direct transmission from the mother to the baby. Another major finding was a reduction in antimicrobial resistance genes. The during COVID group had fewer antimicrobial peptide resistance genes, or AMPs, which help bacteria survive in the gut environment. For instance, we can see in figures 3A and 3B that cathelicidin and defensin resistance genes were significantly lower at 2 to 3, 6, and 12 months. Also, they found fewer antibiotic resistance genes, or ARGs, that protect bacteria from antibiotics. Looking at these figures, specifically 7E through H, there was a significant decline in resistance genes for beta lactams, fluoroquinolones, and rifamycin. An exception that they found was that the Klebsiella pneumoniae had an increased copy number of fluoroquinolone resistance genes, which could be signaling for a potential risk for post pandemic antimicrobial resistance. And this suggests that limited microbial exchange during the pandemic could have weakened the overall microbial resilience of these infants. The researchers also used machine learning to estimate microbiome age, essentially how fast the gut microbiome was developing. And they found that infants born during COVID 19 initially had a microbiome that appeared more mature within the first 100 days, but then their microbial development slowed down significantly at 6 and 12 months. And this means that while some early microbial colonization happened quickly, long term gut development was altered. In addition to changes in bacterial composition, there were also shifts in microbial function. So, pathways that were related to vitamin B biosynthesis, sugar metabolism, and purine nucleotide degradation were reduced. And at the same time, pathways involved in energy production, such as ATP and NADP metabolism, were increased. Possibly as a microbial stress response to the reduced diversity. Um, So, for the discussion, these findings from the study support the hygiene hypothesis, or the idea that excessive cleanliness may reduce microbial diversity and increase immune related diseases. As reaffirmed by an outside source, colonization patterns vary based on birth method, antibiotic exposure, and feeding type, which are all important things to consider because early microbial colonization can influence lifelong health and neurocognitive development. And while hygiene is essential for preventing infections, this study highlights the need for a balance between sanitation and microbial exposure, especially in infants that are still developing and need this for their health. In conclusion, this study provides strong evidence that pandemic related hygiene measures significantly impacted infant gut microbiome development. And while the long term effects remain unknown, it raises important questions about how we should approach hygiene policies, especially for newborns and young developing children. Before we wrap up, I want to discuss some key questions to enable you guys to have some discussion about the article and about the topic in general. How should these findings influence public health recommendations on hygiene? The second question is, how could probiotics or controlled microbial exposure counteract these effects? And what ethical concerns arise? Third question is, what limitations exist in the study design, and how could future research improve on it, in your opinion? And how do these findings resonate with your own experience with COVID 19? And last but not least, what policy changes might be necessary for infant care in hospitals and homes since the start of the pandemic? And here are my outside references. I want to thank you all for your time, and I look forward to hearing your thoughts and discussing these important findings with you. Thank you.